Hi, Gen Bio. This week we are talking about separations, and we are going to look at 3 different techniques in order to achieve separation of different molecules. So the 3 techniques are electrophoresis, chromatography, and spectrophotometry. So the first one that you will use in lab this week is electrophoresis, which is a technique that we use to separate and potentially identify different molecules in solution. So electrophoresis can be used on different types of molecules. We'll be doing DNA electrophoresis, but can also be used to separate proteins or RNA molecules. In any case, the substance is pipetted into a well, which is like a little divot, a little pore in an agarose gel. So agar is used to make a gel and make it sort of a semi-solid structure. And then once you have your DNA pipetted into the well in the gel, which if you look in the picture right here, these are the wells. This blue is the gel itself. Once you have your DNA pipetted into one of the wells, then you put the entire gel into an electrophoresis chamber. And then we can subject it to an electrical current. So anything in that liquid that you pipetted into the well that has a positive charge, that's going to migrate towards the negative pole or the cathode. Anything that has a negative charge to it is going to migrate towards the positive pole or the anode, okay? And DNA has a net negative charge. So it's always going to migrate in this direction from the well down the length of the gel towards the anode, okay? All the pieces of DNA that would be in that mixture would migrate. However, they don't all migrate at the same speed. So any molecule, any DNA fragment that is a smaller size is going to move a little bit faster and therefore a little bit further than the molecules that are larger, than the DNA fragments that have a larger size in terms of base pairs. So those won't migrate as far down the gel. So if you look here in our gel, you can see these little pink bands. The ones that have migrated most during the length of time that you had them in the electrophoresis chamber, those would be the smallest fragments. And then anything that would have only migrated a short distance down the gel, it would have been a larger fragment, okay? And it would be more caught up in the agarose mixture, and so therefore not be able to travel as quickly as the smaller fragments, okay? So let's move to the next slide then. And I'll show you this green is representing our gel again, and the wells where you pipette your substance are at the top, little white rectangles here. You're gonna pipette at least 2 lanes of this gel, so 1 and 2. And the reason that we do that is because in the number 1 lane, you want to run a marker, sometimes called a ladder or a standard. And this is run in 1 lane next to whatever your DNA of interest is in lane 2. The standard then will separate out here. As you can see, there's bands after you've run your gel. You can see that there are different bands that correspond to different fragments of DNA that we know the sizes for. So for this lane, we know what was in that mixture. We know what size DNA fragments were in there. And so I can say, based on information that we would have along with our ladder, what size this fragment is, what size this fragment is, and so forth. And then our DNA of interest would be run in the 2nd lane. You can see this band is representing that, okay? And so we run those next to each other so that we can then compare the sizes. If I know the size of this one and this one, then I can determine the size of mine if I don't know it already, and confirm that it is in fact the DNA fragment that I think it is, okay? So the way that we do this more specifically to make it, instead of just eyeballing it, to make it more quantitative, what we do is we graph on semi-log paper the known sizes of the DNA in our standard or our marker or ladder, whatever you call it. So we're going to graph the sizes that we know that they are against how far they migrate in millimeters. So you will actually take a ruler and measure from the bottom of the well to the middle of the band, and then you will plot that on graph paper, which is actually semi-log paper. Semi-log paper, it just looks like graph paper, except that on the x-axis, you've got evenly spaced lines. You can see that they have an equal size. But on the y-axis, you've got lines that are different sizes. And this is because DNA migrates correspondent to the log of its size. And so when we graph how far the fragment has migrated against its known size, we can get a nice linear shape to our line. Okay, so it'll be nice and straight. And that helps us to determine then the size of the fragment that we don't know for sure. We want to confirm what it is. Okay, so again, we're going to have on our x-axis, we're going to have the distance that each of the bands has migrated in millimeters. And then on the y-axis, we're going to have the known sizes in base pairs for our DNA fragments. Okay, so if we know this one, for instance, is 10 base pairs because it was in my marker solution, and I know that it migrated, say, 80 millimeters from the base of this well down the gel, then I would put a dot here at 10 base pairs and 80 millimeters. And the same for each of the other bands in my gel. I'd plot that on the graph and then draw a line. And then we can use this graph to determine the size of this band. Okay, so that's what we're doing for electrophoresis. The next technique we're going to be looking at in lab this week is chromatography. Chromatography is also used to separate solutes, but based on different qualities. So in electrophoresis, the separation took place based on the size of the fragment. In chromatography, we are going to use paper chromatography, which is going to separate substances based on their polarity. Okay, so again, it's used to separate different solutes in a mixture. There's different solutes. We're going to separate them out so that we can isolate each one. And in chromatography in general, there's always a mobile phase and a stationary phase. For the type of chromatography that we're doing, paper chromatography, the stationary phase is the paper and the solvent is the mobile phase. So we're going to have a liquid that is going to be traveling up the paper. So if you think of it like when you were a kid and you went to a restaurant, maybe you took your straw wrapper and you put it in your glass of water, just like the end of it, and you notice that the liquid from the water would travel up the straw paper, right? I don't know if you ever did this, but my kids do. So this technique is similar to that in that we have paper, ours is cellulose paper, and we are going to have a liquid, a solvent with pigments from plants in it, and that will travel up the paper, okay? So the solutes that we're going to use are going to be extracted from collard greens. So it's a vegetable. We're going to have our lab assistants make this solution up prior to your arrival in lab. So you're going to receive this already made up, but basically it's ground up collard greens, okay, that have been pressed through a strainer so that the chunks have been removed, but you still get a lot of the plant pigments in the solution, okay? So we're going to watch them migrate up the cellulose paper. And what you'll see then is that the different solutes, which would include chlorophyll A, chlorophyll B, xanthophylls, carotenes, different pigments in the collard greens will migrate at different speeds. But in this case, they're migrating based on their polarity and you will see them separate out based on their polarity on the cellulose paper, okay? And just to keep in mind, if you remember back a week or two ago, we had talked about hydrocarbons and how they're made up of just hydrogen and carbon, right? They are relatively non-polar, okay? Elements that have a greater electronegativity, such as oxygen, fluorine, chlorine, etc., they are typically involved in more polar bonds. And so this will lend a polarity or a neutrality to the molecule in general. And so anything that has a higher quantity of polar bonds will be more polar, and anything that's primarily just made up of hydrogen and carbon will be relatively nonpolar, okay? And so based on that quality, then you'll see separation on your cellulose paper. So let's flip to the next slide, okay? So these pictures are basically what you are looking at doing this week in lab. So you have a jar, and the paper we will give you is cellulose paper, and you will see that in the bottom of your jar there's a little bit of fluid, and that fluid is petroleum ether acetone, okay? And that is a non-polar substance, whereas the cellulose paper is a polar substance. And then this whole process hinges on the fact that polar molecules are attracted to other polar molecules. They tend to bind more to more polar molecules. So anything that has a more polar quality to it will adhere more to the cellulose paper, and therefore it will not travel as quickly or as far up the paper as the solvent will. The solvent is relatively nonpolar, and so it will readily move up the paper and separate, and with it will travel the nonpolar molecules that have been extracted from the color greens, okay? So you'll get the most movement of your nonpolar substances and the least movement of your polar substances, okay? Now, the separation of these solutes in paper chromatography can be numerically expressed by what we call an RF factor, or retention factor, which we define as the distance traveled by the solute from the origin over the distance traveled by the solvent from the origin. So for instance, if the solvent travels 10 centimeters up the paper, and you have a solute that you will be able to pick out based on the fact that there's going to be a colored band on the paper, if that solute has traveled 10 millimeters up the paper also, you'll have 10 over 10, which of course equals 1, okay? And so in this experiment, the closer the RF value is to 1, the more nonpolar or hydrophobic that plant pigment is, okay? And the opposite is also true. If you have a number that is much smaller, so for instance, if your plant pigment only traveled 1 centimeter and your solvent traveled 10 centimeters, then you would have 1 divided by 10, which would be 0.1, and that's going to be very close to 0. And that would indicate that it is a more polar substance, okay? So then the RF value will basically indicate the polarity of that molecule, okay? So the different substances that we're separating and calculating RF values for are the photosynthetic pigments that we've taken from the collard greens, and these include chlorophyll A and B, xanthophyll 1 and 2, and carotenes. So you'll probably recognize some of these names, chlorophyll, because that's involved in photosynthesis. You probably heard of that before. The carotenes you might recognize as having an orange color, found heavily in carrots, I believe. So these are the different plant pigments you're going to extract using paper chromatography this week, and we will be calculating the RF values for each of them. Then, after we've done that, calculated the RF values, we're going to do some spectrophotometry. You've already done a good bit of spectrophotometry But up to this point, you've only looked at the absorbance of light at one wavelength for whatever you were analyzing. This week, we're going to look at a range of wavelengths for each of the plant pigments. So we're going to be able to look at then a spectrum of absorbance values that correspond to each of the plant pigments that we take out of the color greens. So then we're going to graph those in Excel and get a nice figure that represents the absorption spectra for each of the plant pigments. And the plant pigments we're not going to do all the ones, we're not going to do all 5 that we look at the RF values for. We're only going to do chlorophyll A, chlorophyll B, and beta carotene, okay? So when you have done that, when you put you'll basically take your paper, your cellulose paper, you'll see a band on it. You're going to cut that band out, physically cut it out, cut it into little strips, and extract it into liquid. It'll elute from the paper, and then you can take the paper out, and the liquid, which then will contain your plant pigment, you can put in the spectrophotometer and analyze the absorbance values. Okay, so once you have done that and you've created your graph in Excel, you should hopefully get something like this picture here. You've got different wavelengths of light on the x-axis that are absorbed at higher quantities by each of the plant pigments or transmitted at higher values by each of the plant pigments, okay? So you'll see a nice absorption spectra similar to this graph right here, okay? All right, last slide. This week's lab, basically what I want you to do is start your chromatography first. You're gonna get that running because that takes some time. And then you're gonna move on to looking at the DNA electrophoresis. Unfortunately, because of COVID, we're under some time constraints. So we're not going to be able to actually run your gel and analyze your results in lab. But what we're going to do is we're going to have sample data, which is already in your lab manual. And you're going to measure and graph the gel bands that are given in your lab manual. So you're going to pretend like you ran it and graph the standard lane, and then use that to compare and determine the size of the DNA in the second lane that we don't know the size for, okay? And then once your chromatography is done running, you're gonna come back to that and you're gonna label your chromatograms. So the paper, you need to mark with a pencil the point at which you started the plant pigments at. So the point which you will put those plant pigments on the paper, and then the solvent front. So how far everything migrated on that paper. And then you're going to mark each of the bands that you'll see on the paper as well, okay? And then once you have calculated the RF values for each of those, then you're going to elute the 3 plant pigments for spectrophotometry and create your graph in Excel of each of the absorption spectra for the 3 that we're looking at, the chlorophyll A, B, and beta carotene. Okay, so that is it for this week's lab. Any questions, you can always shoot me an email at cfry@lvc.edu. All right, have a great week.